Getting that set up, I can start. I wanted to thank the organizers for having me. I think our lab had been doing this work for a little while, and we didn't even have a name for it. And so last year was our first time here, and we appreciate a community that has kind of similar ideas. And I think our work fits nicely into the feelings felt by this group, and so we appreciate being asked to come and give a talk. I wanted to just put no disclosures related to this work, and the main disclosure is that these were the hard-working people that did the work. One of my postdocs, Byung Taek in the lab, Sheng Song, who gave a talk here last year, another postdoc in my lab, and Alexa Livingston, the research assistant in the lab. And just a little bit of outline of what I'll talk about. First, just a little bit of neural recovery. I think we've heard a lot of the same themes through some of the talks here, but just kind of how we came about approaching trying to help the nervous system regenerate and recover. And then a little bit about the tools we use, and then I'll give you two examples in the lab with our animal models in the sciatic nerve and in the stroke model. And I think this audience is well aware how devastating neurologic disease is, both to the patients and their caregivers, as well as a societal burden. As a physician, we always try to learn from our patients. We know that younger patients tend to do better after any neurologic disease, and we've seen that in the lab models as well. And this was just a study done in the 80s by Berganca Sur's lab, who I did a brief postdoc in, where they looked at ferrets and showed essentially how plastic this system can be. And so these are baby ferrets, and if they basically broke the normal pathways to the visual systems of the brain, they showed that if the auditory systems were left open, they could become the visual systems of the brain. So it was a nice demonstration of how plastic this environment can be at certain times. We see it clinically too. This is just one example in the stroke literature of how patients recover from stroke. And the MRS is just the modified Rankin score, since it's a broad audience. Basically, 6 is death, 4 and 5 are inability to walk, and 3 is requiring a lot of help. So basically 0 to 2 is a good outcome, where you have some minimal deficits but can do a lot of what you did before the stroke. And looking across all factors, hypertension, diabetes, size of stroke even, the main one that came out was age. And so you can see if you have a stroke when you're younger, people tended to have a lot more of those 2 and below MRSs than as people got older. So this, I think, presents a nice engineering challenge where how can we recreate that environment to help improve recovery. We also see differently that people change over time. Some people a little more naturally than others, and some a little less naturally. But we know that our brains change over time as well. And so if we can kind of turn back that clock, we may have some success. So as has been mentioned in a lot of the talk, one of the tools we turn to is stem cells and the developing environment of our bodies. And there's been a lot of different talks on different types. So there's the induced pluripotent stem cells, there's the embryonic stem cells, which is mainly what we've used in these studies I'll go over. But I think it's still it's been around now for 20 or plus years, but we're still trying to get a handle on what type of stem cells is best for what application, and our work is continuing to look at that. And just kind of my take on the rehabilitation regenerative approach. If you take a stem cell, I think that gets you a lot of maybe some of the way towards recovery. When we look at it in our neurologic disease models, they're often plopped into unfriendly environments. So stroke, there's a lot of inflammatory changes, peripheral nervous system as well. It's not the most friendly environment for those stem cells. So if you give them a biomaterial scaffold or a boat, maybe they have a little better chance of surviving and doing the work that they can do. And then if you give them a little more guidance with rehabilitation, perhaps, then it's a party, and they can really do the work they want to do. So, that's a little bit how our lab looks at this approach. And so the biomaterials we use to interact with the nervous system are these conductive polymers. There's a lot of different biomaterials that have been used in all sorts of medication or in medicine. to interact with the body. And we use mainly or conductive because of the conductive properties of the nervous system. And the main one that we use is polypyrrole, and it's been around a while in the literature and most used in biomedical applications. The nice part about it is that, as we heard in last night's talk, you can shape things very nicely with the same technology that you make computer chips from with microfabrication. And because you can electroplate this conductive polymer, we can get polymer shapes down to kind of the tens of micron scale to interact with whatever interface we're working on. And so this was kind of some early work just showing that it was very compatible with the brain, had less gliosis than non-conductive materials. And so we kind of have our toolkit now of what we use to work on these difficult problems of regenerating the nervous system. So the first one that was graciously funded by one of the AR3T pilot grants that I'll talk about is a peripheral nerve sciatic nerve transection model project. And damage to the peripheral nerve is a very common problem. Diabetes, also in trauma, in war, or otherwise in motor vehicle accidents. And so a pressing problem. With high cost, and so we need better. Although it regenerates a little on its own, we need better avenues to improve this recovery. And so we were able to make conductive polymer tubes with our polypyrrole, and so these are some images of those tubes. And then with an allogeneic gel, we suspend stem cells in the middle of the tubes, and we can implant them in animals. The first set of experiments we do is kind of seeing how these stem cells just respond to their environment and the electrical stimulation alone. So we're able to see what factors change with the stimulation. And so I think this is a nice component of the platforms we use, is that we have one variable, and if it improves recovery, we can go back to the stem cells and learn what's changing with that one variable, which is electrical stimulation, to give us cues into which path, or clues into which pathways are being modified. And so we've done that in both these systems I'll describe. After, we usually plate the cells on one day, stimulate the next, and then look at what's changed after that. And first of all, of course, we want to make sure we're not killing the cells. And if anything, in most of our platforms, the electrical stimulation seems to maybe increase the numbers a little bit. Definitely percentage death does not increase with the stimulation, so it seems the cells are surviving this. And then we can do some analysis of what is changing, as I mentioned. And so this is some RNA sequencing analysis between the unstimulated groups and the stimulated groups, and you can see there's a multitude of genes that change with this electrical stimulation. And we think of it a little bit like a minor stress to the cells, although it's really not quite understood how electrical fields change the exact genetic expression of cells even today. But it seems again to have a great effect on what the cells are producing, and then we can again dive down into candidate pathways by seeing which genes are changed the most, either upregulated as shown as the orange here, or downregulated as shown as the blue genes here. And then we can take it out to the protein level as well and dive into some pathways. These were just some verifications of common pathways that we know are important in peripheral nerve recovery, such as VEGF-A and BDNF. And you can see after a day of stimulation, both these are upregulated in the stem cells, even before or even just in vitro, and probably similarly in vivo, which we're also looking at now. And so this is our in vivo system. We have, again, a conductive polymer nerve guide here. We do a transection model of the sciatic nerve. And again, the thought process is the stem cells that are producing some nice neurotrophic factors, which we have seen are modulated with electrical stimulation. And then there's obviously the known benefits of electrical stimulation for nerve regeneration on its own. So we have a system kind of combining these two principles. We do some training on our behavior test. We transplant, and then we'll stimulate every other day for about a week after the transplantation. And the idea is that a lot of the embryonic stem cells tend to die off pretty quickly in our system. So it really is kind of what they're producing that's important. And so that's why we kind of stimulate in this more acute time window. And then we'll follow behavior out for 3 weeks with our kind of endpoints, both with behavior test as well as electrophysiology and histology. And so this is just a sampling of some of the behavior we've seen with the electrical stimulation. Sorry, it's a little busy slide, but basically the blue is the stem cells plus electrical stimulation. The orange is the cells alone without electrical stimulation, and then just kind of controls for each measure on its own. This is a test where we place the injured limb down and see how much weight they can hold. And we can score that as well. So this one's actually just the placing and how the paw looks. And so you can see that there's more recovery with this cells plus stimulation group. This is the one where we actually see how much weight the paw can hold. And we also see more improvement in the combined group, maybe starting to see some with the cells alone, but definitely not to the extent. And this is courtesy of Dr. Rando's lab, where we borrow one of their grippers so we can measure how much the animals can grip and pull on a handle, and we see much improvement with the combined therapy as opposed to just individual therapies alone. So this was exciting results, and we're continuing to look at pathways. We also have some electrophysiology we can do in the lab, and these are just some example traces of if we stimulate proximally on the sciatic nerve to our transection and record distally what we're seeing. And again, we really don't see in many of the controls any distal response. And then this is kind of the healthy, normal response we'd expect in a sciatic nerve that hasn't been transected. And we're starting to see a decent amount of recovery at 12 weeks towards similar to that healthy group, and definitely the velocity as well as amplitude. So another marker of recovery. And then we also measure one of the muscles that the sciatic nerve innervates, the gastroc muscle, and see similar improvements with the cells plus ES in just the weight of the muscle, kind of piggybacking on the last talk. If we're using it and recovering the nerve more effectively, the muscle mass tends to stay as well. So another indication that this combined approach seems most effective. And then we're just getting into analyzing the pathology now. So looking at some of these, again, pathways with antibodies, and then kind of the standard, we just histopathology that we see in the tubes without stem cells or electrical stimulation alone. We see kind of a rim of the sciatic nerve that might be kind of hugging the tube that's been described in the literature, whereas with the stem cells, we see much more of a recovering nerve throughout the tube. So the pathology seems to match the functional recovery, which is always good. So that's our approach in the peripheral nervous system. And then moving to the central nervous system, we also have a stroke model in the lab, and stroke is another one of these devastating diseases to the patient, and quite common. And unfortunately, despite all the improvements in the window, patients with other disorders kind of push it off for a while, and they're too late when they get to hospital. So we still see a lot who aren't able to benefit from the acute measures, so we need better recovery options as well. This is just a brief description of our stroke model. Rats are much harder to give strokes than humans, unfortunately. So we basically have to occlude both of the common carotids, which are the main two arteries in the front that come to the brain, and then we do a little craniectomy and permanently occlude a distal MCA, which is one of the big vessels of the front part of the brain, and we get a nice cortical stroke here. And the good part about this model is that you can investigate a lot of the penumbral tissue, so some of this area that people have believed has the most chance of recovery. And so this is the model we used in the lab. And then just an example of the behavior test we do. So because this model sits in the stroke sits in the somatosensory cortex, it interrupts kind of the pathway from sensation of the whiskers of the animal to the paw. And so they have a reflex where if you stimulate their whiskers, they should reach up their paw, which you saw on the unaffected side, they did pretty much every time. And here, after the stroke, you can see BT so here's again the good side, where every time you touch that whisker, he'll reach his paw up to see what's bothering his whisker. And then on the other side that has the stroke in it, there's really none of that reaching up because that signal isn't getting through. And so because it's a little different system, we had to develop a different type of polymer implant. And so here we have one that you can seed the cells on, make sure they're nice and happy on the polymer for a couple days, and then remove it because it's just held together with clamps for transplantation. And so again, the system, like our tubular system, we can investigate how the cells are changing in vitro as well as in vivo. And so this is similar experimental design where we develop our conductive scaffolds. We test and optimize the cells in the chambers, and then we transplant and look at recovery. And this is kind of the general model. Again, we use this Distal MCA model. We train the animals for the behavioral recovery. We give them a stroke on day 0, and then about a week after is when we place the implant in. And then we stimulate this one for 3 days after the implantation and follow out for a few weeks. And this is just, we can do this while the animals are awake, but this is just one where they're sleeping. We've kind of looked at different frequencies and voltages of stimulation to try to optimize that in vitro, and then stimulated in vivo with kind of that optimized 800 millivolt at 100 hertz frequency. And so that's kind of how we often stimulate the animals here. And we found similar results to the peripheral nerve injury, that this combined approach really improves recovery. Most of the stem cell animal literature with this type of model is around here, where, as I mentioned, where they touch their whisker or they touch their paw with the whisker stimulation about 10 out of 10 before they have a stroke. Right after the stroke, they really have no reaches with the paw. And then you can see without any treatment, there's really not much recovery in this model. With stem cells alone, there is some treatment, and usually, again, in the literature, this is about where we get with just stem cells alone, so kind of 5 or 6, 50 to 60% improvement. And then you can see with the combined approach, we're getting very good recovery, up above 80%, 90%. Some animals recover fully, so it's been quite remarkable to see that, the effects of this combined approach. And then looking at why this might happen, which is always our next question, where we see some good improvement. We started to look at the pathology of animals who had this combined stimulation and stem cell treatment, and the brain itself, as other parts of the body, can make some endogenous stem cells, and so it seems really that what the stem cells are secreting is increasing in these endogenous mechanisms of recovery. And so the ventricular zone is one area that's known in the adult brain to make stem cells. Shows if we label for just the dividing cells, which in the brain are mostly stem cells in the adult person, we see with the combined approach there's much more of these tagged dividing cells than just with the stem cells alone. And so we're starting to see some of these endogenous pathways really ramped up with what the stem cells are secreting. And we haven't had time today to talk about. We've done some knockdown experiments with some of the molecules secreted by the stem cells, and we lose this effect. And so again, it's a nice, using kind of this rehabilitation regenerative paradigm, we've been able to kind of zero in on some of these pathways that, there's obviously translating to patients. There's sometimes a higher level if you're trying to get a whole device and transplant in the brain, but now, we have some prospect molecules too that we could perhaps give to patients with their rehabilitation and use maybe the bars a little lower with those. So just in summation, I think as we've heard some previous talks, and I think what's great about this conference is all the different disciplines that are here, but I think biomaterials are a powerful tool to treat some of these neural injuries and get some mechanisms. Again, this combination of approaches, I think, is very important to kind of create this developmental environment where that's very plastic and may lead to improved recovery. And then again, you can kind of read what I've talked about today. So again, just want to thank all the members of my lab group. I'd like to thank Dr. Rando in particular for opening the door to this symposium and group, as well as his help with some of the peripheral nerve injury model, as well as my other collaborators, as well as the AR3T for their support of our work.